Today we have a great speaker. His name is Ian Oglesby. He's a patent agent and pharmacologist. Majored in molecular biology, spent 19 years as a biological researcher in industry, and for the past 5 years has been handling intellectual property for a small biotech company. Please give him a warm welcome.
Well, thanks very much for having me. You got me out of water polo practice, and I appreciate that. I was really excited to see you hear about this series. I'm a big fan of science, always have been. I like to talk about my work, and I thought this was a great way to expose some people to some aspects of science careers that may not be right on your radar right now. Because I'm certainly not doing what I thought I was going to be doing when I was a college student. I always thought I was going to be a college professor. Both my parents are biologists. They're both college professors. Both my dad's parents were biologists. They were school teachers. My mom's parents were also school teachers. I knew what I was going to do. I was going to be a college professor. That changed when I realized I hated academic research when I was in grad school. I'll go into that in a little bit. But, you know, what I want to emphasize with this talk is whatever you do is going to change over the course of your life, and as long as you're looking for opportunities and you keep your eyes open, you can find some really interesting things to do. And I'm going to talk a little bit about that in my biographical part. I got some slides to talk about intellectual property. I have slides that show some of the research projects that I worked on and some projects. I haven't decided how many of these slides I'm going to show. We'll start kind of see how that goes. Like I said, I get very excited about my research and the work I've done. I like talking about it. So I grew up in Southern California. I went to a really big high school. Didn't like it particularly much. Decided to do something, run away from my childhood, so I went to a tiny college in Philadelphia. As far away from my parents as I could get, far away from anyone I knew I couldn't get. Majored in biology. The closest I came to not being a biologist was the semester I thought I was going to be a chemist. And then I took P2x and discovered and kind of reaffirmed that numbers scared me, and I wanted to avoid equations. So I went back to biology because I like mud and squishy things. Haverford, where I went, was a pre-med feeder program. I was the only bio major for my class that was not in medical school within 4 years of graduating. So there were only 22 of us, so it wasn't a big prop. I never wanted to be a doctor. Sick people are gross. And I'm terrified of needles. I worked for a couple of years as a researcher at a hospital in Philadelphia, trying to say, okay, let's see what it's like to actually do research. I really didn't like it, so I decided to go to grad school. I don't know if it was on the introduction, but I am one of the few molecular biologists that has an MA in it. I don't have an MS, I don't have a BS. I have a Master of Arts in Molecular Biology, and that's because where I went for grad school only gave engineers MSs. Even if you majored in physics or comp sci, you would get an MA. Like I said, when I was in grad school, I kind of came to a conclusion that I didn't like academic research. The environment was not very close in my lab. I also made some interesting lifestyle choices. I can tell you it's really hard to study for your master's orals if you have a toddler. This was my first example of that, is taking the opportunity that falls into your lap. My research project for my PhD blew up 4 years into it. I discovered I'd been chasing an artifact for a year and a half. I had nothing. A week later, we discovered we were expecting our second child. I said, cool. I have a really good excuse to leave grad school because I wasn't having fun. Time for me to grow up and support my family. And I convinced my wife that, yes, I was right. The East Coast is a miserable place to live, and we should go back to California. And in a moment, she said, sure. If you really want to be in California again, we can go to California. And a week later, we're driving our little Corolla with a unhappily pregnant wife at the time, because it's really not pleasant to drive across the United States in a week with morning sickness, and a screaming toddler. I learned a skill during this trip, though. I was able to find mashed potatoes to satisfy a craving at 11:30 at night in Muskogee, Oklahoma. And I was really glad I did, because it would have been a bad night if I hadn't. Moved in with my parents, which is also an interesting experience when you have a toddler and a life that's fairly miserable with morning sickness. Luckily, this was in '94, and the job market for biologists was really hot in California then. And I was able to find a job up here within just a couple of weeks. I worked for a little biotech company in Redwood City called Sugen. They're out by the pier where they unload scrap cars and like out in the bay. Nice location. Sugen was an anti-cancer company. It was small, but still private when I started and went public during my 2 years there. And I got my hands dirty in biotech research. I liked it a lot better. My experience for grad school, and obviously every lab is different, every school is different. My experience was that the lab was internally competitive, and that people within the lab in my graduate school were not cooperating. And sometimes they're actually making things harder for each other, and I didn't appreciate that. I'm a very collaborative person. What I found in working for a small company was that it was a very collaborative environment because everybody's trying to solve the same problem. And if you're at a small biotech company, everyone wants to make the next big discovery so they're all rich and famous and can actually buy a house in Silicon Valley. Sugen, like I said, is a cancer company. So we were targeting small solid tumors. That was supposed to be a fairly low hurdle because there aren't very good treatments for a lot of these cancers. Shows the pace of drug discovery. So I was at Sugen from '94 to '96. They were eventually bought by Pharmacia, and Pharmacia was eventually bought by Johnson & Johnson. There are still drugs that I worked on that are in the research pipeline of those companies. You can recognize them because there are numbers that start with SU and then a number after them. SU stands for Sugen from the chemical library. Now, the other risk for working for a small company, of course, is when things go south a little bit, they tend to downsize. So I got laid off, about a third of the company got laid off. That's not a pleasant experience at all. It makes you really doubt yourself. I don't know if you have gone through that process, but after being laid off, they basically told, you know what, we don't need you. You're not worth what you cost to have. That was a sucker punch. And kind of a choice there. There's a series of feelings that you go through. First, you're upset, and then you get angry. And in my case, I decided to wallow for a bit. And my wife has zero tolerance for wallowing when it's in misery, so I was shipped off to the library to start researching other companies very quickly and told I had to send this number of resumes out in this period of time, and I started cracking on it, and I had no excuses to get up off the sofa. That worked. So in '96, it's still a good job market. So within a few, I took a temp job for another small biotech company to tide things over. And then I wound up being hired by Roche here, just down the road a bit in Palo Alto. Roche used to have a big site called Syntax facility right off the page now. Now, like I said, I'm a molecular biologist by training. That's a description of a toolkit that you have. At the time, I saw myself as a set of tools. The department that hired me at Roche was a pharmacology department. At the time, it was called Mechanistic and Neuropharmacology. Our focus was on nervous system-derived diseases of the genitourinary system. I was hired because they needed a molecular biologist, but they didn't have enough work to keep a molecular biologist busy all the time, so, hey, Ian's got little hands, let's do some pharmacology. And that's how I came into pharmacology, was just being thrown at it. I didn't study pharmacology. The idea of measuring the effects of a drug mathematically, looking at efficacy, affinities, and things like that was not part of my research background. So I had to learn. found was very interesting, was a refreshing break. What I found was that, in general, is that molecular biology is very cookbook. You typically are following protocols. What I liked about pharmacology is I was working with living cells. Living cells are, because they're alive, behave or misbehave more often. There's a little more art, it's a little, I would say a little technically harder. I found that kind of refreshing. I got to be involved in a lot of different projects. I'm going to show some, I'm going to show some slides, I think, showing a short pharmacology primer and an experiment I did for a drug series. Now, I mentioned as a general pharmacology department. Once my kids were in school, I started guest speaking at their school, and I learned that you can keep elementary students absolutely wrecked if you talk about pee for an hour. So that's like, that's the guy that talks about pee for an hour. And I can talk about pee for an hour. I'm not going to do it, I'm going to talk about pee for about 5 minutes. So I mentioned the mechanistic and neurogeneral pharmacology department was an umbrella of the neuroscience department. That's what we were. So molecular biology, pharmacology, those are tool sets. That's the tools you have. Neuroscience is the area you study, the features, the aspect of the body, the particular cell types that you're focusing on. Our focus, we had a pain group, analgesia. We had the neurogeneral group. Now, kids like talking about pee and the way the neurogeneral system works because it's got kind of a dirty aspect. It's kind of fun. It's all interesting to talk to adults about because people don't really think about it the same way, especially with the diseases in particular we study. Now, there's incontinence, so both stress and urge incontinence, and I'll define those in a second. My personal disease, when I was considered a disease area lead on, was interstitial cystitis, which is a terribly painful autoimmune disease that's mediated through attack on the neurons the sensory neurons that innervate the bladder. This tying the pain and the general nervous system, there's a reason for it. This is all the peripheral nervous system at work. Now, I want to think about something. When your bladder is filling up, the initial sensation, you start feeling full. That feeling of fullness starts translating into pain, and eventually becomes so painful that you will void. And that, unless you have a serious health problem. In fact, just like breathing, if you fall unconscious, you start breathing. Your body will not let you not breathe if you fall unconscious. Same thing will happen. If you avoid urinating for so long that the pain becomes insurmountable, you will black out and you will wet your hands. So it's kind of interesting phenomenon. The way the nervous system controls this is very interesting. When you think about it, when babies are born, they're incontinent. They void whenever. You gradually master the control of the bladder. That's the central function. That's your CNS taking control of a peripheral nervous function, right? That mechanism, control mechanism is old. It's evolutionary old in mammals. And for example, I think about dogs. Urinating for dogs is an important part of their social interaction. Dogs also use their brains to control what their bladder's doing. That underlying mechanism of voiding without central control is still there with humans. If a person gets paralyzed above the bladder, for the first few days to a week or so, they're going to be catheterized, because otherwise their bladder will fill until they have serious health problems, including death if the bladder ruptures. But after a week or so, they go back to the infantile state, where it's merely a volume level. At a particular volume, the bladder empties. So that's the loss of central control, the periphery can take over again. So what I was interested in, from experimental level, is let's think again about that sensation of the bladder filling, where it starts out feeling like you're filling up, and then it's painful. What we didn't know, and I'm not sure it's still known now because it's been outside my research area for a while, what's happening with the cells that are sending the message to the brain. These are called nociceptors, or sensory afferents. Those are your nerve cells that are telling the brain, hey, I'm full. These cells are very long. One end is in the muscle wall of the bladder. The actual cell body, where the nucleus is tucked into pockets in the sides of your vertebrae. They're called the dorsal root ganglia. And then the other process of the cell is running all the way up through the spinal cord to your brain. What we didn't know was whether this transition from full to pain was a given cell firing more often, so yelling at the brain faster, 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 or was it recruiting other cells so that you have more cells telling the brain, hey, I'm full, hey, I'm full, hey, I'm full. We studied this in a variety of ways. I'm going to talk about it my way in a little bit when I get to the slides, because I realize it's not the first slides of my presentation. But, actually, it might be. Let's see. I'll skip over this and come back to it. Okay. So here's an example of a synapse between two nerve cells. And the molecule, the supermolecule I was talking about in this case is ATP, which is always seen as the energy molecule of the cell. It's what you burn. Every day, your body burns amount of ATP about equivalent to your body weight. More if you exercise vigorously. You're making ATP, continuously breaking it down. Adenosine triphosphates, the energy currency of living tissues. But it is also a signaling molecule between nerve cells. Here's an example where a cell at the top is releasing it, either on purpose through a vesicle or through cell damage. And as ATP can talk to a variety of proteins in the target cell. I'm going to talk about P2x receptors, but there are all these different molecules involved, all these different cells, and various signaling cascades going down to affect the behavior of the target cell. This cartoon's about ATP because I studied ATP signaling. This is true for all neurotransmitters. This is the basic idea is you have one cell talking to another cell chemically. Purinergic P2x receptors. I studied these for most of my time at Roche. I've transitioned off in the last few years. They're ligand-guided ion channels. I'm going to explain that on the next slide. Their endogenous ligand is ATP. So ligand is just a name for the chemical that naturally causes a protein to do something. That's their structure. They have two transmembrane domains, so they're stuck in the membrane of the cell. They have a big extracellular domain that's fairly complicated with disulfide bonds and things. What we know about them, and this is brand new at the time that I was doing this, is that they trimerize. So you get three subunits together. They can either be three of the same or a heteromer where you have two of one and one of another. But within the family, there are actually 7. That's just a cladogram of their relatedness, because they're not that, well, they have a structure. If you could take a picture of one of these, they'd all look very similar, that structure up there. But at the amino acid level, they're not that similar. And as you recall, I talked about dorsal root ganglia. So these receptors, particularly ones that are studied, the P2X3 and the 2,3 heteromer, are on these sensory afferents, extending from the peripheral terminals to the dorsal root ganglia. That thing on the right is the spinal column. This is protein labeling. So we have P2X3 in red, X2 in green, and you can see that some cells are green, they only have one. Some cells are red, they only have the other. Some cells are yellow, that means they have both. This is labeling out in the skin, bladder, various other tissues. And that's where they wind up in the spinal cord, the so-called dorsal horn. These proteins are strictly limited to the peripheral nervous system. They're not found in the brain. That's important because we wanted drugs that wouldn't have effect on the brain if we're targeting the bladder. Sugen was a cancer company. Drugs that treat cancer can afford to have lots of side effects. If you have a cancer that's normally incurable, and somebody stores a drug that will cure your cancer, but make your arms fall off, most people would still take it. They'd rather not have arms and be alive. But for drugs targeting incontinence, the competition is adult diapers. You can't have a serious side effect profile if you're going to be treating it for a drug that's taken for long periods of time. It won't pass the FDA in the first place, but also you won't get patients to take it. Now, the assay I'm going to talk about, it's electrophysiology, which is a pharmacology type assay. It's a functional readout. I'm actually able to measure these P2X channels responding to drugs. It's native, it's not an artificial system. And as far as in vivo target validation, the drug discovery pipeline, at least in traditional pharmaceuticals, is you identify drugs first through testing huge quantities of chemicals in assays that aren't very physiologically relevant. They're usually just measuring affinity for a target protein without seeing whether it does anything. Assays like radioligand binding, where the drug's not contacting anything that's alive. Electrophysiology, the assay I was doing here, this was actually the first test that a potential new drug had where we could find out whether it actually would kill a cell. This is important because really you don't want to waste time and money researching a chemical that will never be a drug. The chemists at Roche got a little scared of me because they'd have these great drugs that just behave beautifully in these earlier assays, and I dropped them on my cells, and congratulations, you invented a really expensive form of bleach. So, as I mentioned, P2x receptors are ligand-gated ion channels. What happens, imagine that little thing's a cell. You have the closed channel, you add the ligand, that causes the proteins in that triplet to change their conformation. You now have a channel in the membrane that allows ions to travel. With P2x receptors, opening the channel leads to a net flow into the cell. It's specific. These guys only will gate for positive ions, and only a few of them. So it's not like you're opening a big hole, letting things run in. This is a process of particular things being allowed through. I was doing what's called a whole cell patch clamp. What I would do is I would make an electrical circuit with an individual cell, hold the voltage difference across the membrane, because I'm not sure what it's not biology, but all cells is a voltage difference between inside and outside, and that's one of the ways cells are able to keep themselves alive and generate energy. I would electronically hold the voltage constant. I could measure the opening and closing of these channels by how much current my machine had to maintain a constant voltage. This allowed me to measure both agonists. An agonist is a drug that mimics the ligand, causes it to open. And an antagonist and agonist prevents the channel from opening or causes it to close. And on the left is a little cartoon of what it looks like. On the right is an actual picture of a cell that's clamped. That would be one of my neurons. I would harvest them acutely and use them fairly quickly because you can't hold the voltage constant once a neuron's extended processes. They need to be rounded up. So this is actually a glass capillary tube that I've pulled out to a very small diameter. You can see how small that tip is relative to the cell. There's an electrode in it. If you look in this cartoon over here, the voltage electrode's in that glass tube patch of cell is reference electrode out of the cell. The cell's sitting in a bath, and I can add drugs to the solution that the cell's sitting in. And this shows the current traces as you get close to the capillary tube. That's the resistance drops. In panel C, I have now fused with the membrane, and we have those transient spikes. I zero them out, and then suck a little bit of pressure. Actually, we're going to do this using a syringe and using my mouth. Just suck a little bit of air and pop a hole in the cell. Give those large spikes, and then I would zero those out. And no, it doesn't happen on every cell. This is a very frustrating process. When I first started doing electrophysiology, it took me 6 months to not kill a cell trying to do this, which did not make my boss happy at all. Now, so in this particular set experiments, the ligand that I was using is alpha-beta methylene ATP. Basically, it has, if you look at the structure of ATP, we have the triphosphates. This had methyl groups replacing them, so it's non-hydrolyzable. cells use ATP for energy by breaking off the phosphate groups. You couldn't break off the phosphate groups with alpha-beta-methylene ATP. The reason we use this is because the responses to the different receptors that I was studying was different. I could tell them apart. ATP can't tell them apart. Okay, so the quick experimental paradigm, patch a cell. Okay, takes, you said that. Add a little alpha-beta-methylene ATP. I should see the channel open and see a current, an inward current. Wash it out. I mean, I keep doing that until the current that I see stays the same, because I'm looking for a difference. So I can't, if the cell's being variable, I can't use it. Then I would just put the drug on board for a couple of minutes, wash that out so there's no more drug present in the well, add the alpha-beta-methylene ATP again, and what I'm looking for, if the experiment works, and this would be one data point, is if the peak from the D step was within 15% of the peak from the B step. That's a sign that I'm actually measuring a reproducible phenomenon. This is what I'll be seeing on my oscilloscope. For, you can't see the bottom. The horizontal scale is milliseconds, and the vertical scale is picoamps. I add drug at that second blue bar. There's a slight lag because it has to diffuse through the basins of the cell. The channel opens, slowly starts closing, and then turns off when I remove the drug. So it's a very fast response. When the ligand is there, it's open. When the ligand washes off, it's gone. This is what a P2x 2-3 pentamer looks like. This is what a P2x 3 homomer looks like. You can see the response shape is different. It opens, and then on its own it turns off. The drug is still present. That's a very sharp peak because the channel just opens, quick signal, and done. Trinitrothenol ATP is an antagonist. It competes but does not open the channel. It's fairly efficacious. It has a 10 to the minus 7th molar TNP ATP, completely eradicates the response to 10 millimolar alpha-beta-methylene ATP. So that'd be one data point. But it's all as a screen. On the left, you see it's an EC50. It's the dose response to alpha-beta-methylene ATP. That's moles on the bottom, and it's percent response on the left. On the right shows what screening of an antagonist looks like. So in this case, I'm giving the cells alpha-beta-methylene ATP, and then in the presence or absence of this drug, and it shows a nice dose response. So the EC50 is about 10 to the minus 7th for turning off the receptor, preventing the receptor from opening. Flipper stands for fluorescent light imaging plate reader. It's one of these endpoint assays where the cell dies. I would do correlations of my data versus Flipper data. We want them to match fairly well. If they don't match, we have to figure out why. DRG, dorsal root ganglia, those are the ones that I talked about, were in the pockets of the vertebra. No-dose ganglia, they only express P2X2. They are at where the vagus nerve enters the brain. And really quickly on this, because this is taking longer for the EFIS that I was planning on, question of whether a particular drug is acting competitive or non-competitive. So an antagonist can work through one of two mechanisms. A competitive antagonist prevents the agonist or the ligand from binding to the same thing, but not causing the physiological effect. So it's competing for a binding site. A non-competitive antagonist binds somewhere else in the protein and prevents the signal from being propagated, either from changing the conformation of the protein so that the agonist can't bind, or through some other mechanism. They can be distinguished pharmacologically in a couple ways. A competitive antagonist, if you add enough of the agonist, you'll block the antagonism. You just swamp it out. You just overcome it through simple kinetics. A non-competitive antagonist, you can't. It's going to knock down the ability of the agonist to do anything, because the agonist and the antagonist are not competing for the same site. this is a little test with surmountability. If it's competitive, you can surmount it by adding more of the agonist. And in this case, for this particular drug candidate, it did. This is about three weeks to a month's worth of data, because this was N's of 5 for each of these experiments. It shows what the throughput is for this type of assay. So in this case, the first challenge is 10 micromolar ATP. Add drug, and you notice the response drops. Now, then I add 30 micromolar ATP, and the response back up where it was at 10 without drug. At 100, I'm even higher, and then then 3 challenges with 10 and drug, I'm back down to where I was, and then I can recapitulate the ATP response there. So what this data shows is that this drug was acting competitively, because by adding enough of the agonist, the antagonist could no longer block the agonist action. I'll wait on that for a second. So I was at Roche doing this kind of stuff for a long time. I realized after a while, it took a while, I was enjoying what I was doing, but I kind of wanted to do something interesting, in something a little different. Now, I had fallen into another example of things of serendipity. I had gotten to meet some people through Little League Baseball and Boy Scouts. And this is a husband-wife team that had started an artificial intelligence company. And they had a project for DARPA, which is it's a branch of the Department of Defense, which basically they give money to people with crazy ideas on the off chance that something will pan out someday. What DARPA was interested in is, could you use a predictive model looking at chemical structures to prevent side effects or toxicity without actually having to test the molecules in either animals or people? This was for the biological warfare aspect. They want to be able to know if they found it, what's this, what will it do? They could find out rapidly without having to go through elaborate testing. That was the project. My friends asked me to be along because they were not pharmacologists, and I was there. My role was to be the idiot checker, because I couldn't understand their tool at all, but I could tell whether their results were making sense or not. And that was a fun thing to do, and it went on for several years. Got some papers out of it, got to give some interesting talks. Had to get a security clearance, so that was kind of exciting to find out from my friends that had been funded by the government. That also led me to thinking that I've been doing research for a long time, kind of wanted something else. What else can I do without going back to school? So I knew I didn't want to go back to school. The idea of, I still have, I don't know how many of you have this nightmare. You think you're awake and you realize you haven't studied all semester, you haven't done any of the homework all semester, and it's the final, and you don't know where the classroom is. I still have that nightmare. I could not go back to school. Patents. Now, I got interested in patents actually at Roche, because part of my little group, I had several RAs. One of the drug targets we were studying was a membrane receptor, not P2x. And one day, Merck got issued a patent on that receptor. And we got told, okay, we're done. And that was 7 man-years of work done. We got a couple publications out of it, but the compounds just went away. Weren't in this time. That was kind of a shock to me. Okay, what is this? What is this powerful thing that can scare a drug company off from pursuing a research project that actually was going someplace? Patents. Now, this is from a Supreme Court decision this summer. I picked it because it's particularly important relevant to my work at Clontech, where I am now. Myriad Genetics is a company that got patents on the sequence of a couple of genes implicated in breast cancer. But the sequences they patented were the sequences that are in our bodies. And they controlled the testing market for diseases. They were charging several $1,000s to test for diseases. BRCA1 and A2. Not all breast cancer are related to these genes, but people that have women that have particular mutations in one or more of either of those genes have a greater than 50% chance of getting breast cancer. And frequently people are diagnosed with one of having mastectomies just as a preventive measure, because they're that predictive of cancer. And Myriad was charging $1,000 a test, and they were sued on the grounds that you can't patent this. This isn't a man-made thing, this is a product of nature. And what this has been in the courts for 10 or 12 years, and the US Supreme Court finally decided this summer that the myriad patents are invalid because they're the actual sequence that's in our bodies. But they also held that recombinant DNA, cDNA, can be patented because that's a product of man, which is the requirement for getting a patent. You can't get a patent on a law of nature or a mathematical algorithm. You can only get a patent on something that required a creative input of a human being. So I decided I was going to be interested in finding what I could do involving patents. Now my speech, and I'm going to go back to school, is to be a patent agent, which is what I am, or a patent examiner, which are the government employees that kind of handle the other end of the argument. You actually don't need to go to law school. All you have to do for a case of patent agent is pass the patent bar exam. So that's what I did. I studied for the test, and it was a miserable, awful test, hardest thing I've ever done, but I didn't have to go back to school to get a law degree. That's why I'm a patent agent and not a patent attorney. If I had a law degree, I'd be a patent attorney. The requirement, this is one reason I brought up for this talk, the requirement for being a patent agent or a patent examiner is that you have to have a degree in a STEM field. You know, you have to have a certain body of technological knowledge. They're not going to be they won't be hiring people that don't have a scientific background or value a patent. I got really lucky again with this transition because Roche decided to get rid of my department. I found this out in February of 2007, but they told me I was going to be laid off. I didn't know what day I was going to be laid off until April. And then they kept extending it. So I wound up, they didn't have very much work for me to do. I was the last person in my department. I was the last person in my building, which is both kind of fun, but also kind of scary. I was able to go for really long runs, and nobody knew. But when I'm sitting on my EFIS rig, not moving very often, the lights are on motion sensors, and I'd be really focused, and all the lights would turn off, and I didn't have any windows, so it got really, really dark in the lab. So I learned to periodically wave an arm up in the air to keep the lights on. But the nice thing was I could study for the patent bar, because I didn't have a lot of free time on my hands. I don't think I would have been able to study for the patent bar. You know, kids at home and everything like that, there's no way I was going to study at home, so I was able to get through it. Passed the bar. Made a discovery. If you're a biologist, everyone wants you to have a PhD to be a patent agent or a patent attorney. CS, CE, all the engineering fields, bachelor's is good enough. But for biology and chemistry now, it's pretty much a PhD thing. So eventually Roche did get rid of me, and I was job hunting, and I was getting a lot of rejection letters. So I got depressed. So okay, I have to actually, it's not just send pajamas in front of my computer and spam law firms with my resume. So I took a job doing tech support as a temp for Kelly Scientific, and they gave me Clontech. And I was there a few weeks, and I mentioned to my boss that I was a patent agent. He said, "Oh, what's that?" And I explained to him. And the next day I became Clontech's intellectual property department. They hired me full-time. They don't have a full-time IP department. I do intellectual property, patent work 2 days a week, and I still am in the marketing department doing tech support 2 days a week, and doing other marketing on other days. So I can work on product literature, rewrite manuals, deal with customer issues, selling calls, and things like that. Okay, I'm going to rush through this because I can talk about patents as long as I can talk about P. But what I wanted to do this should be, I think this is an interesting slide. I want to talk about intellectual property in general. And this goes beyond, this is applicable to everybody, not just people from a scientific background. So a patent is a piece of property. It's a license from the government. It basically says, you have the permission to do this thing, you and only you, for a period of time. But in return, you have to tell everybody else how to do it so they can improve upon it. So it's a complete description. That's what it actually means. It means open letters. It's property. You can buy it, you can sell it, you can lease it, you can give it away, you can do what have you, but it's property. The other forms of intellectual property in the US and in most of the rest of the world, is a trade secret. The trade secret is the opposite of a patent. A trade secret, as long as you don't tell anybody what it is, it's yours forever. You never have to give up a trade secret. If somebody finds out what it is, it becomes public knowledge, you've lost it forever, even if it's not your fault. Industrial espionage, going after trade secrets, gets people, makes them open game. What I recommend at my work at Clontech is, if the thing you're selling, you can tell what your invention is, well, you better get a patent on it. But if the thing you're selling, you couldn't tell what it is, maybe it was a process by which you made something, then you're better off keeping it as a trade secret because it lasts longer. A trademark is a non-descriptive name for a product. It's yours forever. In the US, you can actually just have it as long as nobody else tries to keep it away from you. But it has to be non-descriptive. Clontech sells a PCR, preliminary chain reaction enzyme, we call titanium. And we tried to get a trademark on it, and the US Patent Office, the Trademark Office came back and said, well, you can't get a patent on titanium because it's made out of titanium. And we had to write back, no, it's a protein. There's no titanium there at all. Eventually we got it. A copyright protects expression of ideas, but imitation or parody is allowed. You have a copyright automatically, here's me write down. If you take notes and talk, you have a copyright on your notes. You can pay to protect a copyright, but you don't have to. You'll notice here, one thing I think is kind of important to think about, especially if you're interested in computers, especially in software. There is not a good place to stick software in the structure of intellectual property law. Right now, software is patented, but in order to get patents, since you can't patent an algorithm, software patents are worded very weirdly. Usually they have to say somewhere in the claims that it causes electrons to shoot across a screen or causes a button to be pressed. It has to do something. Copyright seems like a more obvious spot for software. The problem is, copyright works really well for the written word, where if you wrote something different, it'd be different. It's a protection against plagiarism. With software, there's lots of different, you put two programmers on, write a program that does X. The chances they're actually going to come up with the same code is very small. So copyright can't cover it. It's not, plagiarism really is an issue, it's look and feel. I won't go into this, like I said, I can go into patents for a long time. But I think we're getting time where I should ask for questions. We have, I want to put this picture up. This shows a couple of Clontech products at work. Those are nerve cells. And they've been infected with lentiviruses, HIV-1 to be exact. Very interesting lentiviruses. They don't carry viral coding, viral genes. What they did carry was a gene for a fluorescent protein. One is ACGFP1, comes from jellyfish, Aquaria ceruleans. Other are DS-RIG, which comes from a coral named Discosoma. The ACGFP1 has been fused to actin, which is part of the skeleton of a cell. You can see that looks like fibers running through, especially the left-hand one. That's the structure of a cell, the skeleton of a cell, blood cells, they're in that shape. Whereas the nuke, there's a nuclear localization signal that targets that protein to the nucleus of the cell. So you get these nice, pretty pictures that really illuminate the structure of a cell. And Clontech cell, these are Clontech fluorescent proteins. We have a huge list of colors. If you Google glowfish, there's a company that sells fluorescent fish for the pet market. They're illegal in California because they can live in the wild here. You can buy them in the rest of the US, but they license our fluorescent proteins. So if you live in New Jersey, you can buy a Dayglow pink fish or a Dayglow blue. There's going to be a company fairly soon as we're releasing fluorescent rodents for the pet market. So if you really want to have a bright orange mouse, especially one that glows really brightly under dark light, you're going to buy one. And really quickly, I just want to show how what we did for the lentivirus. So that's a cartoon of what of HIV-1. The only parts of HIV-1 that are still there are the so-called long-term repeats and that funny Greek letter thing. It's the phi packaging signal. We cut out the middle, which had all the genes for HIV-1. We just kept elements that would make, you'll make a variant. This variant can infect the cell, but it cannot reproduce because it doesn't have its own proteins. Within that, we have various elements for expressing another gene, a transgene. So this is where they're introducing foreign DNA to a cell. What I like about this cartoon shows a couple things. It shows the beauty of evolution. It also shows what the phenomenal things that you can get together with science and by the way things work. Because this is a chimera of so many different things in the natural world. Like the WPRE. I didn't know that woodchucks could get hepatitis, but this is from a virus that causes hepatitis in woodchucks. It also happens to promote RNA processing in a cell. The CMV promoter, that's that black box up there, that's cytomegalovirus. That's a human virus. It's disease-carrying in people who are immunocompromised. Most people are carriers with no symptoms. A promoter is an element that makes a gene fire. The PGK is phosphoglycerate kinase promoter. That's one of ours. That's a human promoter. And inflammasome resistance, which is a protein in bacteria that provides antibiotic resistance. So this is a chimera of things from every kingdom of life, even non-life, since viruses aren't technically alive. I think I'm going to stop here because I know there's hopefully some questions. I could prattle on for quite some time, but I don't want to run overtime on this. But if anyone would like some, ask some questions, I'd be glad to answer either now or after the talk's done. So thank you very much.
Do you think the current U.S. patent system has a lot of vulnerabilities being used?
Yes. And it's always being tweaked. The problem is most of the tweaks, they just change, they change the vulnerabilities. I think things have improved in biology a lot. 10 or 15 years ago, companies were getting patents just on DNA sequences like that Myriad patent. They didn't have to know what they did. You just got a patent on a sequence. And companies pretty much claim the entire human genome. Those are now gone. The Supreme Court decision said, no, that's ridiculous. At the very least, you have to know what this thing does. It's more a question more of the way some companies can use patents to bludgeon other companies, either in trolling or just through intimidation. It's still a problem. Clontech is a small company. We sell tools to biologists. We're a reagent company. We're not a drug company. We can't afford to be sued or to sue anybody. So there are companies that are selling our fluorescent proteins. We have patents on fluorescent proteins. All we can do is write letters that say, hey, either give us money or stop it. But we don't have a stick to back that up with. So yes, I think there are real vulnerabilities. I think protecting intellectual property is so important because I do believe it encourages creativity because it gives people the opportunity to profit from their inventions, and that's the purpose of it. But yeah, it's definitely areas of abuse. A couple things have been fixed recently that I really like. False marking. It used to be you could sue a company for false marking even if the product had an expired patent number on the packaging. And the fine was like $750 per. Well, we had a company troll our website, download our expired user manuals, and then threaten to sue us for all the old patent numbers in them. And the thing is, they could tell how many times these manuals have been downloaded. So $750 a pop when a manual's been downloaded 120,000 times, and my company's revenue is $22 million a year. That was a really scary letter. We were very happy when the government changed that rule.
Any more questions?
Yeah.
I grew up in Philadelphia, so nice to meet you. Okay, my first question is, do you have autonomy when you work for a big pharma company? And my second question is, what does your boss specialize in?
Well, when I was at Roche, my level of autonomy varied with time. I had actually quite a bit because I usually was pretty good at being the only person who knew how to do what I did. I was very lucky at Roche. It was a great company to work for. I don't have a PhD. I was able to prove myself and got promoted into a PhD level position. And so I got through a glass ceiling and I was encouraged. So from my experience at Roche, and again at Clontech, I think it was, I had quite a bit of autonomy. At Roche, Roche's bio was scientists all the way up, so my boss was also a pharmacologist. Although it was interesting, my boss for much of my time at Roche had actually started out as a cage watcher in the animal facility. He had an AA degree in environmental science, and he became a department head because he was that good at his job. Roche allowed that. They don't do it anymore. Roche has changed. They're a lot more sclerotic. At Clontech, I have two bosses. One is he's a scientist. He's basically R&D head and business development. And he's from Oxford. He's British. He sounds like a Monty Python character, so he's fun to talk with. My other boss is a chemist by training, but has been in marketing. He's the head of marketing. He's been in marketing most of his career. So both non-scientists and science, both from a science-y type background.
Any other questions about how he managed to navigate this path or any of the science stuff? Really? It says woodchuck hepatitis up there and no one has a question? There we go. I may have opened a can of worms with that one.
Speak of like the woodchucks. How much would a woodchuck chuck? Okay, no, I can't. I'm sorry. I don't want but it's actually kind of amusing to find that woodchucks could also get this hepatitis. And does it also pass on to humans, or is that completely?
No, it's a distinct disease. I'm not sure it's in the same family as either of any of the viruses that cause hepatitis in humans. I really have no idea. It's kind of a tricky question. And my response to the first part of the question is, how much ground round would a groundhog hog or groundhog could hog ground round?
Groundhog Day.
So I mean, these were built. These were built. The question is, why would anyone do this? As far as why would you put these things together? It just shows what a lot of people can do. I mean, that's the great thing about science. It's collaborative. To make something like this took hundreds of different people, because different elements are discovered by different people, and it's putting them together. That's one of the things I love about being working in the science area. You know, so here's a paper I'm on, back to your responses, and you look at, you know, how many people are on that and how many different places they actually worked. This is for one paper, not a very big paper in a sense, it's a fairly small piece of information. I really like that about science, that we have people here, now most of all these addresses at this time were either in Britain or in the US, but countries of origin, going from on the top or left to right, we have Ireland, China, Australia, Germany, Mexico. Second row is England, California, Germany, England. It's a great thing about science. Yeah.
What was the most unusual project you worked on, or at least one of your companies worked on?
Most unusual. I mean, certain piece kind of unusual, but we had an interesting thing come up for a program I was only briefly affiliated with, and so at Roche we're studying one of your serotonin receptors. This is for single nervous systems, and we're getting some really bizarre results in the experiments that were done on living cells. Things that are supposedly agonists were not doing their full signal. And what I realized, and it took a while to figure this out, you know, when the light bulb goes over ahead, it's like, we should have thought before. Cells that are grown in culture are grown typically in 10% serum. Usually it's fetal calf serum, sometimes it's horse serum. Well, all mammals have serotonin. It's in the serum. Basically, we were bathing our target in the endogenous ligand, and then trying to discover how to turn it on. It was a set up for failure. It took a while. It was kind of a backwards way to find that out. It was a very interesting little duh moment. All right. Thanks very much.